techniques for examining the dynamics of proteins and other biological molecules in the glassy sugar matrices commonly used for their preservation.
Language. But I will say I'm really pleased and honored to be able to stand up here and talk about Mark a little bit and how deserving he is of this award. So I want to say that many people start their careers at NIST, coming straight in from their degrees and PhDs and going to NIST and their postdocs. And Mark did sort of a circuitous route to NIST. He went to industry first, where he worked in orthoclinical diagnostics. And then he moved to academia, and he taught at Brigham Young University. And then he came to NIST, and we were lucky enough to get him in here at NIST for the last 14 years. His experience with industry, I think, is evident because his work is very problem-focused, and he really always seems to be able to anticipate the needs of the community. And I think that's really something a lot of scientists sort of lose sight of. His experience in academia is evident, and that he has a real knack for teaching science to really all levels. And we have kids that come through, we also have congressmen that come through. Wait, that's the same level. Okay, let's and we have other people come through who are esteemed scientists. And he can talk to all of them and teach them about science. It's really wonderful and amazing. And at NIST, he's really excelled in all aspects of his job, performing groundbreaking research and mentoring the next generation of students and postdocs and scientists, and then translating his work into the community. It's really amazing to watch. So tonight, the Washington Academy of Sciences is recognizing Mark with the award in the biological sciences in recognition of his transformative research, and I mean transformative research, in high-impact optical measurement methods for quantifying biological systems. And I'll tell you what these things are used for. Most prominently, he's recognized for having established this new type of spectroscopy, and I'll call it BCARS. It was spelled out in longer words there. And basically, it enables direct chemical imaging of cells and tissues. And his work involved really innovative, creative design and engineering of broadband laser sources, detection schemes, and methods. And all of these were aimed at really doing high-resolution, chemically resolved images of living cells and tissues. And with this, you can look for things like, look at cells and look at them to analyze for disease state. And that's really a profound thing with a labelless technique like BCARS. His achievement really opens the door to things like better cancer diagnosis and tissue engineering, and then new advances in precision medicine, all very exciting fields. He also established new methods for looking at the dynamics of biological molecules and the way that they're commonly preserved. And this work has a direct impact on the design of preservation schemes that help maintain safe storage and transport and reliability of things like vaccines, and also these new classes of engineered drugs like protein therapeutics. He's truly a leader in his field, and his research has amazing and broad influence and impact. I've always had such a pleasure to work around Mark, to know him. Somebody walked up to me in the hall and said, "I've never met Mark Cecirone, can you describe him to me?" And I said, "He's a superstar." So I'm really proud of Mark's work, proud to know him, proud to work with him, and congratulations for your very deserving award.
I came to NIST about 15 years ago and started working, although it was my first opportunity to work with biologists, I did start working with biologists. My background is in physical sciences, and it really cemented the idea to me that folks doing biology have a very, very difficult problem. They have very complicated systems, and it's really hard to get quantitative, reliable information to work with. And so I thought there must be some way to extract from these cells and the tissues that these guys are working with quantitative information, and perhaps in a label-free way. And so thought about this a little bit. when folks are looking at tissues and cells and they're trying to manipulate them and send them down certain pathways and get them to differentiate and function in certain ways, what they're really asking them to do is to generate certain chemicals at certain times. And so what we're really looking for was chemical changes in biological cells. And so being a physical chemist, I thought spectroscopy would be the way to do this. Every type of molecule has its own vibrational fingerprint. And if you could just pick those fingerprints up from the cells and the tissues that you're working with, you could learn what in fact they're doing. And I wasn't the first person to think of this. Folks have been doing vibrational spectroscopy on biological systems for decades. In fact, an interesting little tidbit, for some types of cancer, for kind of marginal cases, depending on which doctor you go to, you've got about a 50% chance of getting the right prognosis and the right diagnosis from a histopathology, from a tissue slice of the cancer, or not cancer, from the tumor. So with really advanced cases, it's a little better, 80%, but for marginal cases, it really is about a 50% drop. However, if you use vibrational spectroscopy to look at what chemical species are in these tumors, you actually get a 95% correct prognosis, or diagnosis rather. The thing is that physicians don't use vibrational spectroscopy because you can't do it in an imaging modality. It takes a second or up to 10 seconds to get a single good quality vibrational spectrum using standard techniques. And physicians want images, so they want really millions of these individual pixels. And that's just not been possible. So about 10 years ago, I imagined maybe there was a way to get these vibrational spectra more quickly. And so I'm not going to tell you about the 10 years because it was mostly grinding. But now we have a technique that in just a few milliseconds we get these full vibrational spectra where we can identify individual molecular types within these samples. And so now within milliseconds. So in just a few minutes I can get really nice, high quality images that tell all about the chemical composition of tissues and cells. It's exactly the information that you need to get that 95% diagnosis, but now we can do it in minutes. The program is starting to get recognized. I think that we're really just at the very beginning. We're now on the millisecond time scale getting these spectra. I think we're going to go to tens of microseconds this year, and I think that we'll have something exciting pretty soon. So, thank you so much for the award, for the Academy for the award. It was really a complete surprise to me. I really am honored and appreciate it.